All right, so this topic is going to be about hormone actions at its target cell. And that is specifically related to once a hormone is secreted from its gland, it enters the body fluids. Specifically, here's how it, let me get a board up really quickly. Here's the basic gist of what happens. If this is can be any hormone gland whatsoever, endocrine gland, it synthesizes the hormone inside of these cells inside of the gland. The actual hormone is just a small chemical messenger. It gets made or synthesized in here and then secreted from these cells. And it depends on which gland, the various means of secretion, but ultimately that hormone makes its way out of the endocrine gland cells into the interstitial fluid. And then if there's a blood, typically a blood capillary, a really small blood vessel, that hormone, I'm just going to put an H here, makes its way into the blood. And then that hormone circulates everywhere in the human body and is exposed to all of our tissues. But then only the cells in the tissues that have some sort of a receptor for that hormone, will that hormone actually act? So, and we're going to talk about this process here at the target cell. So that's most of today's conversation. So I'm just going to erase this and then quickly go to a new board. So this is called hormone actions at the target cell. Hormone actions at the target cell. What happens once the hormone finds its target, basically, is what we're talking about. First, when they reach their target cell, hormones typically alter some sort of, I'm going to put this in quotes, activity at the target cell. And we'll look at some of those activities as well. So let's do that now. I'm just going to give you some examples. Examples of altering activity at the target cell would be things like they might change permeability of the plasma membrane at the target cell. So once they reach the target cell, they sometimes alter the permeability of that target cell's membrane to allow some things to come in, or perhaps preventing some things to come in, whatever that may be. They may also stimulate enzyme synthesis and activity. Or they might activate an enzyme once they get there. An inactive state of an enzyme might be at the target cell. When the hormone reaches the target cell and binds to it, that might, in quotes, turn on that enzyme to do something. Hormones can also induce secretions at that target cell. Might cause the target cell to secrete something, might even be secreting another hormone. And sometimes hormones, when they reach their target cell, can stimulate mitosis, which is just cellular division. So I'm just going to remind you, I know that most of you have not had a full Bio 1 class, if ever, then for a very long time. And cellular division means typically increase in tissue growth. So those are just some examples of hormones, how they might alter that activity at the target cell. So I'm going to layer over this and just say the hormones act at their receptor site or at their target cell basically in one of two ways. And it's based on what we talked about yesterday, the chemical composition of the class of hormones. So remember yesterday we did two classes of hormones, amino acid-based hormones and steroids or steroid-based hormones. That chemical structure class dictates pretty much how it behaves at the target cell. So I'm just going to write, now we're going to get more specific. Hormones, we're still talking about what they do when they reach the target cell. And this is based on I'm going to write chemical class. And what I mean by that is whether or not they're, that's CL. Let me just correct that, make that look a little better. This means based on whether or not they're amino acid in chemical structure or lipid-based, steroid-based from cholesterol. So I'm just going to label this number 1 and 2 just like we did yesterday with chemical class. And number 1, I'm going to write water soluble hormones, which are amino acid based. So what this is saying is that all amino acid based hormones, which are also water soluble, interact at their target cell in the way that I'm going to describe. There's an exception to this. So I'm going to write that first. The exception to this rule is thyroid hormone. Thyroid hormone is amino acid based, but it does not behave this way at the target cell. All other hormones that are amino acid based interact with their target cell in this fashion. So I'm going to write a few words and then draw a really nice picture. So here's what they do. The hormones act on receptors in the plasma membrane of the target cell. So that's first and foremost. Second thing I'm going to write is the receptor. That means this receptor is coupled with a G protein. I'll explain this in pictorial form after we write these couple sentences. So the receptor is coupled with a G protein, and we call this whole process, so I'm going to write process, it's referred to as a second messenger system. All right, so those are just some important keys to remember as I describe this process. So let me just layer on top of this, and we'll go to a pictorial form of explanation. So these are water-soluble, I'm just going to title my diagram here, water-soluble hormones. Second messenger system. Now I'm going to draw a really large cell. And this would be the target cell. Okay. And then I'm going to draw a hormone. And their hormones are very small. So I'm going to draw a hormone out here. The hormone is happy because it's the last day of class. So here's our hormone, and just remember this is amino acid lipid-based hormone. That's its chemical structural class. Now, as we said yesterday, and this is key to remember, these amino acid-based hormones are water soluble. The plasma membrane of almost all of our cells in the human body, it's called phospholipid bilayer. And the key to remember there is that they are lipid-based membranes in our cells in our body. We know that water and fat, water and lipid, don't mix. So this hormone, because it's water soluble, it can't penetrate this membrane because it's lipid-based, it's fat-based, so it gets repelled. Therefore, the target cell for this hormone has to put a receptor out here on its surface that's made specifically for that hormone. So it can't just go into the cell. So it needs to bind to a receptor on the target cell's surface. So I'm going to draw that. There's our hormone. It's bound to a receptor that's designed specifically just for that hormone. So this hormone right here is circulating everywhere in the human body through the blood. When it reaches or finds this target cell that has this specifically made receptor for it, it will bind to it. Now, this receptor, we're going to get into why this is called a second messenger system. This receptor is coupled with a G protein. Now, there are G proteins in this cell, on the inside of this cell. When this hormone binds to its receptor on the outside of the cell, that sort of activates this G protein that's coupled with it. I don't have any room to write, so I'm going to write some things at the end of this, but I'm going to say this now. This G protein is inactive when GDP is bound to it. And don't worry about what GDP means. It's guanine diphosphate. But when the hormone binds to the receptor that's actually physically coupled with this protein, it attaches a GTP to this G protein, and then that activates it. So just think of this. Hormone binds to receptor. Because this receptor is physically coupled with the G protein, it activates this G protein inside. And then that activates something called adenylate cyclase, which is an enzyme. So I'm going to draw this. I'm going to draw an enzyme attached to the G protein, and I'm going to write adenylate cyclase. That is an enzyme that gets activated by the G protein being activated. Now, this adenylate cyclase, when activated, I'm going to draw an arrow this way. This is all explained in your book, but if you watch me do it, hopefully it'll be a little more simplified and clear. So when it's a cascade, it's called a cascading environment or event in which we activate one thing and it activates another and another and another. So this binding out here activates the G protein, which activates adenylate cyclase enzyme. And once it's activated, it converts ATP in the cell, that same molecule that we talked about a long time ago, to something called cyclic AMP. So I'm going to write little c. I'm not going to test you on what all of these abbreviations stand for. I will say them, but it doesn't mean you have to actually. Cyclic AMP just means cyclic adenosine monophosphate. And I'm not going to test you on this part, but ATP has 3 phosphates. It's called triphosphate. And this adenylate cyclase cleaves off not just the terminal phosphate, but actually 2 phosphates to make it cyclic adenosine monophosphate. What I do want you to know that this cyclic AMP is designated as the second messenger in this system. The first messenger is actually the hormone. So there's our first messenger that was released from some endocrine gland. It travels through the blood, binds to its receptor. Then we have a cascading event of activating a G protein, which activates adenylate cyclase, which is an enzyme that converts ATP to cyclic AMP. This is cyclic AMP is our second messenger in this second messenger system. And then cyclic AMP activates, I know it's crazy complicated, but this is what happens in our cells every day, activates kinases. That's just a class of chemicals, of enzymes that typically phosphorylate proteins. They add phosphates to things. So kinases, I'll write what they do here. They phosphorylate proteins inside of the cell, and that usually increases the cellular metabolism. It can also inhibit, so it can crank up the cellular metabolism, or it can actually turn it down, depending on this long process, what gets activated and what doesn't. So I'm going to go through this really quickly once again. Here's our hormone. It's a amino acid based hormone. It cannot enter the cell directly because it's water based and our plasma membrane is lipid based. So it has to bind to a surface receptor on the target cell. When it does that, it activates this G protein, which activates adenylate cyclase, which is an enzyme that converts ATP to cyclic AMP. Cyclic AMP is our second messenger that activates kinases in the cell. Kinases, that's just a class of chemicals, of intracellular chemicals that typically phosphorylate proteins, which ultimately usually ramp up or increase metabolism of the cell. That can go up or down, increase or decrease, depending on the target cell. So a G protein or protein kinases are just these things. These kinases are just really enzymes that phosphorylate proteins. That is a classic example that I've drawn of a second messenger system. Believe it or not, this isn't the only second messenger system that exists. So I'm not going to go through all of the intricacies of the other classic example, which is calcium, but I'm just going to add on to this. I won't erase it, and just say a few more things. So we know that a G protein can activate adenylate cyclase. We just went through that. I'm just going to say, or this is just another example of a second messenger system. Or a G protein can release calcium ion stores from inside of the cell, and then I'll put this in parentheses. Calcium can activate something called calmodulin. That's supposed to be, I'm getting ahead of myself on the spelling. That's calmodulin. Calcium can activate calmodulin, which just activates enzymes. The reason I'm writing this out is this is this second classical second messenger system. what I want you to remind is that I want to remind you is that calcium can act as a second messenger as well. So that's the other classic second messenger system in the human body at the target cell, is that calcium gets released after the hormone binds to the G protein. Calcium gets released inside of the cell, and it's calcium ions that act as a second messenger. And they can do the same thing. They can alter cellular metabolism. All right, let's leave this now and go to the second type of interaction at a target cell by a hormone. And this is going to be number 2, steroids. I'm going to put in parentheses, and thyroid hormone. So thyroid hormone is technically not a steroid. It's actually amino acid based, but it's so small it can get through, and it's also based on its chemical makeup. Even though it has amino acid bases in it, it can also behave just like a steroid. It does not behave like a typical amino acid based hormone. So let's do the same thing. Let's write this. Usually what happens with a steroid is we get direct gene activation via intracellular receptors. Okay, so now I'm going to draw my big picture again, my target cell. And we're going to watch how a steroid behaves. So our steroid hormone, I'm going to make it a different shape out here. Let's make it a triangle instead of a circle. But the steroid is still happy because it's the last day of class. Now, this target cell, it's just a cell in the human body. It has a lipid-based plasma membrane. But guess what? Steroids are lipid-based because they're derived from cholesterol. So lipids, fats, and fats can mix. And the key number one difference from an amino acid-based hormone is that steroids, because they are derived from cholesterol and are lipid-based, they can slide right through this plasma membrane. That's a key difference. This is our steroid hormone. It does not bind to a surface receptor on the target cell. It enters the cell directly. Now, in this target cell, because all of our cells have a nucleus, I'm going to draw a nucleus, a really big one, and I'll label that. There's our nucleus of the target cell. Once this steroid enters the target cell, and it does so directly, right through the plasma membrane, it can bind to a cytoplasmic receptor inside of the cell. So I'm going to label this. So there's the receptor. It's not out here on the membrane because it doesn't have to be. It's actually in the cytoplasm. So we have the steroid hormone entering directly, binding to this cytoplasmic receptor. Now we have a hormone receptor complex. Inside of the cell. And that hormone receptor complex enters the nucleus of the cell. I'm going to make it a little smaller. And here's typically, we know what's inside of the nucleus: DNA, genetic material. So I'm going to draw some DNA in here. There it is. This hormone receptor complex enters the nucleus and binds to a specific section of DNA. And the binding of this hormone receptor complex to a very specific piece of DNA, what it does is it initiates the process of protein synthesis. So over here on the side margin, I'm going to say the hormone receptor complex enters the nucleus. It binds to a specific section of DNA. When it does that, it initiates protein synthesis in the cell. This is now, all steroids don't act this way, don't have this specific action. This is just a very common example. So what we're doing is, we are actually, I'm putting this in quotes, we're turning on a gene to make a specific protein. And you know the rest of this story from the first week of class. Once this hormone receptor complex, I'm going to use different colors just so you know where I am on the drawing. This hormone receptor complex in here binds to DNA. When it does that, guess what? The DNA uncoils. Then we can make an RNA. From that, the RNA molecule gets modified to messenger RNA. The RNA leaves the cell. I'm just going through this process that you already know. The RNA goes to a ribosome. Here's our ribosome. And then that RNA message that was copied from the DNA gets read or translated through the ribosome, and we start to make a protein. So I'm just going to write. So that whole process that you learned the first week of class of protein synthesis, this is what initiates, this is a big factor, one of the big factors that can initiate protein synthesis. It's a steroid hormone. You've heard of anabolic steroids, right? They cause people's muscles to grow or get bigger. Guess what? That is initiating protein synthesis in our cells to make more muscular tissue. So that's a very typical example of a steroid hormone and how they behave at the target cell. So I am going to ask you all sorts of questions about these two processes, the differences, what they do, and what I'm going to leave with, the last thing I'm going to talk about is 3 types of endocrine gland stimuli. So we're off of this target cell business right now. So I'm going to layer over this board, go to the next one, and just write 3 types. I believe this is the last section that we've assigned in the textbook. Of endocrine gland stimuli. We're at the opposite end of the pathway here. What I mean by, we just talked about what does a hormone do when it reaches its target cell. And we gave 2 classic examples of a water-based, amino acid-based hormone and a steroid, what they do when they get to their target cell. This last topic, it just refers to, I'm going to go back to my very first picture, which looks like I've erased, so I won't. Remember when I talked about what does an endocrine gland do when it first secretes a hormone, when it synthesizes and secretes its hormone and releases it into a blood vessel? That's what this topic is about. So we're actually at the endocrine gland itself, and we're talking about how it makes what stimulates it to synthesize and secrete a hormone. So we are at the endocrine gland, not the target cell. And there are basically three major types of stimuli. First one is known as, that's a U, H-U-M-O-R-A-L, humoral stimuli. You and I know the word humor as funny, haha, humor, but the Latin-based word humor means body fluids. So humor, when we, in A and P, I'm gonna write humor equals bodily fluids. Really, it means this is a very simple form of endocrine gland stimulation. When we say bodily fluids, we mostly mean the bloodstream, so I'll add that. Obviously, the blood is not the only place where we have fluid, but that's where we have most things in a fluid, suspended in a fluid-based medium. So what happens is, I'm going to write from levels of certain, say, ions or nutrients. This is the cause of endocrine secretion. This is very simple to understand. All we're saying is this. Certain levels of ions or nutrition, whatever, certain levels of certain things in the bloodstream dictate whether or not a hormone, an endocrine gland is going to secrete a hormone, okay? Classic examples of this would be, I'll write some examples. Calcium levels in the blood. This is, I'm going to use this one first because we know about this. We know that 99.8 or whatever percent or more percent of our calcium is stored in our bones, and there's a very small level that's constantly circulating in our blood. But those levels in the blood are very strictly monitored. If they rise or decrease, something happens. And we learned a long time ago that a decrease in those blood calcium levels, even a tiny bit, stimulates the release of parathyroid hormone from the parathyroid gland. And then that stimulates an increase in calcium levels in the blood. So just decreasing calcium levels in the blood stimulates, so let's just say this is a parathyroid gland, it stimulates this gland to secrete parathyroid hormone. And then the actions of this hormone causes an increase in blood calcium levels. So that's a classic example of humoral stimuli. Another example, this will also be fairly easy to understand. Glucose levels, sugar levels in the blood. When we get a decrease, sorry, I'm going to erase that. When we get an increase in blood glucose levels, that stimulates the pancreas to secrete insulin. insulin gets sugar out of our bloodstream and into our cells. It gets that glucose across our cellular membranes. These glucose levels in the blood are also strictly monitored. If they rise, insulin is secreted from the pancreas. If they decrease, glucagon is secreted from the pancreas. So that's another classic example. And finally, a third classic example is aldosterone release, and that's based on sodium and potassium blood levels. So this first example of humoral stimuli just means levels of something in the blood can stimulate this endocrine gland or any endocrine gland to secrete a specific hormone. So that's number one. There's just three and then we're done. Number two, endocrine glands can be stimulated by neural stimuli. So our nervous system can also stimulate, that's supposed to be an I, can stimulate an endocrine gland to secrete a hormone. So it's very straightforward. Nerve fibers releasing neurotransmitter onto an endocrine gland, stimulate endocrine gland to synthesize and secrete a hormone. Example, here's a classic example. When we are stressed out, now you know that stress activates the sympathetic nervous system. And those sympathetic fibers tell or signal the adrenal medulla. The adrenal glands are just located right next to or above our kidneys. They signal the adrenal medulla to secrete epinephrine and norepinephrine. You and I know those two hormones as adrenaline. And that does all sorts of those sympathetic effects on the body, like increase heart rate, dilate bronchioles. Finally, number three, the third thing that can stimulate an endocrine gland to synthesize and release a hormone is a hormone itself. So we have covered humoral stimuli, just means the level of something in the blood. Neural stimuli, nerve fibers can act on an endocrine gland to synthesize and release a hormone. And finally, another hormone can do that. So this is just hormones stimulating the release of another hormone. So when one hormone binds to its surface receptor on a cell and it changes that cell's metabolism, then that cell then synthesizes and secretes another hormone and releases it. Classic examples. The hypothalamus secretes those releasing and inhibiting hormones onto the pituitary gland. and then it tells the pituitary to synthesize and secrete its own hormones. So it's just one hormone causing the synthesis and secretion of another hormone. Last thing, last bit, I'm going to just make a start here and make this statement. The autonomic nervous system that we just got finished studying really has the ability to override the endocrine system. So the nervous system can ultimately override and control the endocrine system in many different ways, usually in extreme circumstances, like stress, for example, causing shutting down an endocrine gland or enhancing it. So I'm just going to write turning off endocrine secretions or enhancing them, turning them up. All right, guys, we have reached the end. I'm going to stop there because that's what I said I would do on the outline. So let me turn off my recording.